طيب, so this is the second lecture about translation. So I think we went over all of this, صحيح? Last time we reached to a point where we talked about the initiation factors, which are proteins that are involved in recruiting وbinding the small subunit of a ribosome على the beginning of the mRNA. And we said even the end of the mRNA plays a role in recruiting هذا الcomplex for preparation and initiation of translation, صح? Where مثلا the poly-A-tail binding protein is involved مع إنه binds على RNA region that's all the way at the end. Furthermore, this slide is talking about an element, a sequence, which is known as internal ribosome entry site, IRES. That sequence, بالإيكاريوت, it recruits or binds the initiation factors, بتعرفوا عليه initiation factors, sequence معين, and based on it, the translation machinery is recruited as well. For not only the 5' cap will poly A-tail, rather the UTR itself is involved, صح ولا؟ لأن if you look على ال IRAS هذا, it's upstream of ال AUG. بما إنه before the AUG, معناه it's part of the 5' UTR. So this shows you and encourage you to appreciate ال UTR الرول تبعها here بالtranslation. صحيح؟ So what's written? خلينا نشوف شو مكتوب هون. ال IRAS exists in some gene mRNAs of certain genes recognized by 40S اللي هو هذا اللي هون ribosome or initiation factor for G protein. As you can see هيو هذا ال pink اللي attached على green site اللي هي element followed by recruitment of a small subunit. With initiation of translation, this slide is straightforward in biology 101 where the small subunit attached to it the mRNA وبعدين the start codon recognized by the initiator tRNA, which is a tRNA carrying the methionine. This in turn, it's GTP driven in order to assemble the large subunit al complex هذا. Now the AUG, the start codon is recognized by a tRNA carrying methionine, and in bacteria, most bacteria it is methionine بس عليها الكربون هاي الالديهايد group هاي, so we call it formyl methionine, not methionine. Yeah, nothing complicated here. The three phases, you are aware of them, and we mentioned them over and over previously: initiation, elongation, and termination. And they require factors of proteins to assist in them. على سبيل المثال, بالprokaryotes, there is initiation factors 1, 2, and 3. Those initiation factors, they play a role in docking the small subunit. هون شوف عندي هون عندي الinitiator tRNA that's carrying الفورميل ميثايونين والانتيكودون تبع الاي يو جي اللي هو السي اي يو. هذول الانيشيشن فاكتور بالحاله هاي اي اف 2 مع جي تي بي is binding الانيشيتور تي ار ان اي على الستارت كودون and other initiator factors اللي هم 2 3 1 and 3 they bind السمول subunit تبعت الرايبوسوم على الانيشيشن ريجن. وبعدين once the whole ribosome مع ال mRNA مع الinitiator tRNA are together, they dissociate. So to sum it up, initiation factor 2 plays a role in recruiting initiator tRNA. Initiation factors 1 and 3, they play a role in recruiting the small subunit. Pay attention when we're talking about bacteria, so small subunit هي 30S والlarge subunit هي 50S, whereas in eukaryotes هم 40 and 60 respectively. اللي بيعملوا كل اليونت هي 80 صح؟ بينما بالإيكاريوتس هي 70. وهاي هي الستبس تاعون GTP driven, you have to know that, where the charge tRNA بالelongation binds على A site, amino acid tRNA binding site, وبعدين the ester bond is being broken, we're forming a peptide bond, and the elongation continues. And this is the machinery. طبعا the peptide bond occurs between the growing polypeptide على C terminus, بصير addition of the new amino acid من the charged tRNA, وهذا dehydration reaction that forms the peptide bond. And this is an animation shows the process that is ongoing during elongation. Now the stop codon, what binds to stop codon, not tRNA, rather issue اسمه release factor. وهذا الريليز factor will dissociate the tRNA from ribosome, therefore ribosome subunits والmRNA will eventually separate from each other, وهون بصير عندي termination of translation. لاحظوا بعد الستوب كودون بعد الريليز factor there is still mRNA, and this region we call it UTR, 3' UTR, after the stop codon. Further, moving further, it's telling you in prokaryotes, the genetic material, DNA, is in a region, not a compartment. And higher region is in a nucleoid. Therefore, as the DNA is transcribed to RNA, هذا الRNA لسه عماله being released from the DNA, being transcribed by the ribosome. So transcription with translation, they came at the same, occur at the same time, while in eukaryotes and the compartmental nucleus and the membrane envelope, two membranes actually. So therefore transcription occurs, RNA processing, and then export the cytosol, the mRNA translation. Pay attention to the fact, in a sense, the mRNA, يعني as it's being transcribed, it's being translated, معناه there are no introns and exons بالRNA تبع البكتيريا, right? There is no timeless spliceosome يجي remove the introns. So the concept of introns does not exist in the prokaryotes. لا كل الMRNA له stop codons, yes, كل الMRNA they have stop codons. البوليزوم والبولyribosome, it's the idea that multiple ribosomes are attached to the same mRNA at the same time, making multiple polypeptides right after each other. Well, look at this picture. This is the DNA, and here the mRNA is released from the DNA, where ribosomes are attached to the mRNA in the transmission electron microscope. This is prokaryote ولا eukaryote? Prokaryote, لأنه transcription and translation occurring at the same time. Furthermore, do you think the initiation of transcription up ولا down نبلش؟ The process of transcription نبلش من فوق لتحت ولا من تحت لفوق؟ من فوق لتحت. صح ليش؟ لأنه look الmRNA اللي هون أكبر من الmRNA اللي هون, right? So this is the polysome or the polyribosome, the same meaning they encompass. For moving further, I want to discuss with you another point اللي هي cloning. Now listen to me. Cloning معناها إنه the same organism or the same cell to be duplicated. يعني أنا إذا بدي أنسخ حالي, make a copy of myself, يطلع واحد تاني carrying my same DNA, بعتبره my clone. أو bacteria, I put a bacteria on a petri dish and I allow this bacteria to divide through binary fission. والباينري fission بعطيني daughter cells carrying the same genetic material, لأنه الباينري fission identical DNA duplication, صح؟ زي الميتوسيس. فالبكتيريا daughter cells and the daughter cells من original one bacteria, a group of cells هاي bacteria, they're considered as a colony. Why? لأنه if I look at their DNA, it is almost identical, وأصلهم من one خلية بتطلع similar cells. هذا cloning. Another definition of the cloning بمعنى آخر هو إنه so that in the future if you read, it means إنه if you want to identify where a gene, find a gene and identify where it's located on a chromosome, كمان بستعمل كلمة cloning. If I'm telling you someone is cloning a gene and someone is trying to identify a gene and where it's located in the chromosome, two different meanings for the same word. هذا بالإنجليزي word زي present معناها حاضر أو هدية. طيب, another term that you should be aware of اللي هو transformation. Transformation, we talked about it, and we said it's the process of a normal cell turning into a cancerous cell. تحول الخلية normal لكانسر, هاي البروسيس بسميها transformation. Another term encompass معنى آخر للtransformation هو when I'm telling you I'm transforming a bacteria with a DNA. Introducing a foreign DNA into a bacterial cell كمان هذا المصطلح بستعمل transformation. كلمة معنيين مختلفين كليا. واحدة إنه to introduce DNA غريب عن البكتيريا جوا البكتيريا، والثانية إنه تحول normal cell لcancer cell. والterm is well established. Transformation. Let's talk about the cloning. البكتيريا it has a chromosome. Some of them have 2 chromosomes. يعني على سبيل المثال البروسيلا has 2 chromosomes. another bacteria tuberculosis تبعت التوبركلوسيس هاي 1 chromosome فيها. غير الكروموسوم aside of that some bacteria عندهم extra genetic material. عندها circular double stranded DNA we call it plasmid. شو اسمه؟ plasmid. شو هو؟ هو extra genetic material. What does كلمة extra imply? It implies the fact إنه I can remove this plasmid in bacteria and it can live on طبيعي. لأنها extra. So what happens? This plasmid I can cut it certain restriction enzyme. From the human, for instance, gene الانسولين I cut the ends in another tube and test the tube في البلازميد تبع البكتيريا. بحكي I can call a company بحكي لهم listen بعتوا لي البلازميد اسمه PRP23 في لهم اسماء موديلات مختلفة. They send me in a tube. بحط فيه restriction enzyme. تيوب تاني في gene of interest مثلا الانسولين. I cut that gene with the same restriction enzyme. So the ends of تبعون الجين cut with the same restriction enzyme that the plasmid is cut with. بعدين I mix them together in one tube so the ends are compatible. None of they are cut with the same restriction enzyme. صح ولا لا؟ So if you add ligase it will induce the formation of covalent bonds between الستراند اللي تحت والستراند اللي فوق. And it will carry, now I will end up having circular DNA, part of it from the bacteria, part of it from my gene of interest, the human gene. I call this DNA recombinant DNA. You are expected to know what recombinant DNA means. This is recombinant DNA. Bacterial DNA عليه human gene, for instance. And then I transform the bacteria with this recombinant plasmid. And the transformation procedure you are not required بالامتحان, but basically you create transient temporary pores in the bacteria. إما خلال electrical current تضربها electricity بيفتح pores بالبكتيريا، أو بتعملها permeable من خلال chemical treatment not electrical treatment. بحط مثلا معها MgCl2. البكتيريا becomes permeable. I add the plasmid إلها. It goes in. Now I have a bacteria. Inside of it is the plasmid. بحطها على petri dish and I leave it to incubate. راح تعمل binary fission. Identical daughter cells بيحمل نفس البلازميد. بعدين الكولوني هاي I take it and I throw it in a big flask فيه media, liquid media, يعني nutrients for the bacteria, and it will keep dividing binary fission. وصار huge quantity of bacteria كلها colony من single bacteria صار فيها البلازميد, and then that bacteria is transcribing and translating the gene of interest تبعي. So and then I harvest the protein من البكتيريا as a therapeutic protein. Now that's the recombinant DNA in the context of يعني producing protein, therapeutic protein زي الانسولين. Furthermore, some proteins are more complicated. بمعنى ممكن some of them عندهم tertiary structure, multiple subunits. So post-translational processing تبعهم is complicated. أو كمان في شيء اسمه post-translational modifications بنحط عليهم chemical groups like methyl groups أو acetyl groups أو sugars. So but the bacteria sometimes fails. It can give you a polypeptide, but it's one thing عندك polypeptide. Another thing that polypeptide إنه يحصل على secondary, tertiary, and quaternary structure and becomes a functional protein. And what's a functional protein? هو three-dimensional structure. البروتين اللي تعريفه هو functional polypeptide. So if the protein is complicated, يعني إذا ظهر بالevolution later on بالmammals والvertebrates حجمه كبير, maybe the bacteria will fail to give you the proper folded protein. Then the system هذا you can use other cells other than bacteria to transform. You can use yeast. You can use, if it's more complicated than yeast, you can use إشي أقرب, إشي within the animal kingdom زي insect cells. Or if the protein insect cells بدش, they produce, transcribe, translate. هم incubator كبير as a matter of fact. Or you can use mammalian cells that are capable of expressing the gene of interest. مش جوا المايس in vitro بالفلاسك. ممكن someone might ask hold on طب ليش نعمل ببكتيريا؟ ليش ما من أول هنعمل mammalian cells وريح راسنا؟ Maintaining the mammalian cells is much more expensive with the much more sophisticated equipment. Well, rate of division تبع cells is way much slower than bacterial binary fission. That's why you start with a simple cell, simple system to express your gene of interest. If it fulfills the requirements, good. If not, you upgrade. معلومة إذا صار مش مطلوبة بينكم الامتحان. It's one thing, the gene of interest, to express it in a certain type of cells, prokaryotic or eukaryotic, and it's another thing to use a virus. الفيروس موضوع مختلف كليا. What a virus is, and I did that يعني for many years. So بنجيب adenovirus. الادنوفيروس هو اللي بيعمل الفلو فيروس الرشحة. And then we design it, we remove a replication gene so that when you infect the patient with 1000 viral particles بضلهم 1000 viral particles. And before that كمان we introduce a normal gene, the DNA of a normal gene inside the DNA تبع الفيروس. And then we try experiments هاي. We are not allowed نجرب على human مباشرة. Rather we have to do preclinical studies. The preclinical studies معناتها in vitro and in vivo. So in vitro يعني in the dish نجيب cancer cells من patients and I used, so what we did إنه الفيروس هذا carrying a gene, that gene when expressed الفيروس بدخل جوا الخلية and then it expresses the gene. Well, the product of the virus induces the cancer cell to die but not the normal cells. البروتين تبعه. It worked perfectly great. عمل cells, the cancer cells بيكون على petri dish attached على surface تبعه, and when they die they will shrink وبصيروا floating بيسبحوا ميتين وفي media liquid على cells. Now and it worked very very well. Breast, prostate, colorectal. We tried it on a battery of different cancer types and it worked fine. Next step, try it in animal in vitro, in vivo. جبنا المايس. We injected the mice with human cancer cells. Those human cancer cells, already inside of them there is a gene تبع اللوسيفريز, الفروت, الفايرفلاي. في دبانة firefly عندها gene اسمه لوسيفريز بضوي. So the idea the cancer cells, the human cancer cells, before ما أدخلهم على المايس بحط فيهم اللوسيفريز وبعدين بدخلهم بالسيستم تاع المايس. So I can image المايس تحت جهاز معين أشوف الكانسر وين وجوده وكيف انتشاره, how it spreads, how it grows. بعدين we injected المايس بالفيروس هذا. Well, الليفر كان مشكلة عويصة. الليفر هو الجريفيارد اللي بيعمل كابتشر للفيروسات. It filtered الفيروس, منع الفيروسات يمشي بالبلاد circulation. I said okay, let's try to bypass the liver. Skip the liver. وبعدين مشكلة تانية صارت. الimmune system is recognizing الادنوفيروس نفسه as a foreign body and it's attacking it. The immune system تبع الانيمالز ارتفع. So the point, الثيرابي في الكانسر مش الاكتشوال efficacy المشكلة هي الدليفري والتارجتنج, يوصل الثيرابي تاعك للتيشو of interest. That's what's challenging.